It's a pleasure to be here. I'm an associate professor at the University of Minnesota, also now in the position of senior scientific advisor at Diversigen. Was the CEO of CAZyme, which has merged into Diversigen. I'm going to tee up Eric a little bit, who's going to talk about some new analysis methods that have been put in place and are being made available at Diversigen as a service for analyzing carbohydrate-active enzymes. And I'm going to tee it up just by talking about a study that we did recently in my academic lab in which we found these quite useful. And this is going to be in the area of obesity and US immigrant health. I'm sure many of you have seen these plots of the incidence rate of metabolic diseases, not to mention all of the autoimmune disorders skyrocketing all over the world over the last like 50 years, but primarily in industrialized nations. And actually, what I hadn't realized is that the same thing happens on a much shorter time scale for groups of people who move to an industrialized nation from a recently developed nation. And we can see this across many different US immigrant and refugee groups where the rate of obesity and being overweight, it rises almost a factor of 2 within just a few years of arrival in the US. So it seems that although most immigrant and refugee groups arrive in the US generally metabolically healthy, within a short period of time, they take on the same risk of obesity as others in the US. And we had a big question about this in my research lab. Is this driven primarily by diet, or could the microbiome be playing a role? And so we ran the study that was led by Pajao Vange, who was a PhD student in my lab. And Pajao is ethnically Hmong and worked with the local Hmong community. Then we also worked with the Karen community, which is another group of refugees. We used a community-based research process, which was fabulous. I highly recommend it. It let us bring these groups into the design of the project and make sure that what we did resonated with them culturally and was interesting to them, personally. So we try to ask the question, what happens when a person moves from here, this is a refugee camp in northern Thailand, to suburban Minnesota or urban Minnesota? What happens to the microbiome? And how is that related to obesity, and how is that related to diet? To do this, we had to acquire samples from people still living in Thailand from these 2 different ethnic groups. And then from first generation immigrants, so people who had moved to the US in their lifetime. And then we also have samples, these are stool samples, from second generation immigrants who are of the same ethnic group, but who were actually born in the US. And then finally, at the far end of the spectrum, we have US controls. These are white European Americans or people of European descent who've been in the US for multiple generations. And we also did a little bit of longitudinal sampling. So we started off with these big picture analyses using 16S sequencing. And we can do all of the standard things like alpha diversity, beta diversity. And this started to tell us an interesting story. So in beta diversity, we saw in this principal coordinates analysis that the Thailand residents of these 2 ethnic groups, the Hmong and the Karen, they were kind of in their own sector of microbiome space, and they tended to be fairly consistent within each group. Then so those are on the left. On the far right, we have the US controls. The first generation immigrants are everybody in the background, some of whom more resemble pre-immigration or people still in Thailand, some of whom are more like the US controls. And then finally, on the right, also on the right, we have the second generation immigrants. And what this is showing is that after a generation in the US, people's microbiomes tended to be almost indistinguishable from other people who had been in the US for multiple generations. But first generation seemed to be in this kind of transition state. So we looked at, and from this we concluded that US immigration westernizes or industrializes the human gut microbiome. So then we looked at phylogenetic diversity. And here we saw in Thailand that the diversity, phylogenetic diversity was fairly high. This is not terribly surprising because we've seen this in other studies where in more recently developed nations and also in more rural settings, people have more gut diversity. But then, and I apologize, the colors didn't come out in the conversion to PDF here. But these are actually lean and obese. So people who were in Thailand but had obesity had a slightly lower diversity than their lean counterparts. Then we have first generation lean. And this shows that just moving to the US causes a larger drop in diversity in the microbiome. Well, we think it's a cause, you know, here it's just an association. A lower drop in diversity than obesity. Then we have obese first generation, lean second generation, obese second generation. And there seems to be this step function where the largest driver is where you live and your immigration status, and then obesity is another driver. And by the time people are in second generation, just like with beta diversity, their alpha diversity is more or less in line with US controls. So this is a little bit of evidence for this, what's been called the disappearing microbiota theory, which says that as people are exposed to industrialized society, antibiotics, lack of fiber, perhaps environmental pollutants, formula feeding instead of breastfeeding, all of these cause us to lose microbes, which we then can't pass on to our kids. And then they have fewer microbes to pass on to their kids and so on. And so this is a little bit of evidence in an actual multi-generational cohort that we see the effect of industrialized society on the microbiome. But none of this told us anything about the functional changes. What was going on under the hood in the microbiome? We know they're losing a lot of native taxa and picking up US taxa. What's happening to the genes? And this is where we turned to shotgun sequencing on a subset of 55 individuals. And we showed we used this deep shotgun sequencing. It was about 25 million reads per sample, all on stool samples, to characterize the carbohydrate-active enzymes using the CAZyme database. This is what Eric is about to talk about. So I'm showing on the vertical axis the different CAZymes, and then the horizontal axis is people. On the left are the Hmong still in Thailand, and you see they have a high abundance of all of these purple colored enzymes at the bottom and not many of the ones at the top. Now on the right, these are US controls, so they have almost a completely different set of enzymes. And then in the middle, these are first generation Hmong who've been in the US for a long time, at least 10 years. And this is where we could see functionally that whatever the microbiome was degrading and consuming in terms of fiber and complex carbohydrates in Thailand seems to have changed, and people's microbiomes seem to have almost completely lost those carbohydrate-active enzymes that were highly prevalent in Thailand in favor of these ones that are highly prevalent in the US. And it turns out these are actually related to, they seem to be related to foods that are more frequently consumed in Southeast Asia, like palm, coconut, konjac, tamarind. And this took a lot of work, some fine tuning, you know, it was kind of a one-off analysis that we did. So I'm gonna hand it over to Eric, who can talk about the implementations and kind of scalable, automated way that Diversigen is starting to do this.
All right, thanks, Dan. Hi everyone. I'm Eric Smith. I'm a bioinformatician at Diversigen, and like Dan said, I'm going to tell you about a database that I've helped develop and the service that we're launching to profile CAZyme content of metagenomic samples. And before I get too far into it, I want to acknowledge that a lot of this work was done as part of an ongoing collaboration that we at Diversigen have with Prescient Metabiomics. And they were pretty critical in funding a lot of the work I'm going to tell you, and sort of were the driving force behind developing this database. So just so we're all on the same page, I want to give a brief intro to what CAZymes are. So as Dan mentioned a few times, CAZymes are carbohydrate-active enzymes. So they're any enzyme that's involved in any sort of modification of carbohydrates, whether that be breakdown or building up carbohydrates or adding on functional decorations or anything like that. And they're split into sort of 5 major classes. There's a couple minor classes that I'm not going to get into great detail here. I'm also not going to get into great detail about specifically any of these classes, but just wanted to list them there for you. And then within each class, I've also listed the number of families that that class is broken up into. And then within those families, in some instances, they're also broken up into smaller subfamilies. And in addition to those 5 major classes, there's a website called CAZy.org, which is really a very informative website that has pretty much any information you could be looking for on CAZymes. They've also annotated what they call carbohydrate binding modules. So these are non-catalytic, and they are often tagged on to CAZymes, and they help with adhesion to carbohydrates and binding the substrate that the CAZyme acts on. So just some brief information about why they're important. So as I've said, they're involved in all aspects of carbohydrate biochemistry. A lot of plant pathogens carry a repertoire of different CAZymes to break down plant cell walls and plant-derived carbohydrates. So here's a study that looked at a plant pathogenic fungus and found that it has a suite of CAZymes that are induced in the presence of plant-derived carbohydrates. Many CAZymes play roles in the digestion of carbohydrates, so in the healthy human gut. So here's a study that profiled the CAZyme content of the healthy human microbiome and how it utilized CAZymes and complex carbohydrates. Here's CAZymes might impact the structure of the digestive tract mucosal lining. And as you can imagine, that's sort of the first line of defense against invading pathogens from the gut. So there are implications for disease there. So here's a study that looked at diets that are deprived of fiber shift the microbiome in such a way that these CAZymes that degrade the mucus barrier of the colon increase, and that enhances pathogen susceptibility. And then they might also be important for processing biomass for biofuel production. So here's another study that identified a CAZyme cassette that is useful for degrading biomass in thermophilic bacteria. So how CAZymes have traditionally been annotated. So each family that I talked about is based on at least one biochemically characterized enzyme, and then other enzymes are added to the family primarily based on sequence similarity, which is determined using hidden Markov models. And so this involves assembly and gene prediction, and then running the predicted genes against these hidden Markov models to determine CAZyme content. And this number is a little outdated, so CAZy.org also actually has closer to 18,000 bacterial genomes that have been annotated for CAZyme content now. They also have some archaea and some eukaryotes identified. But what we wanted to do was to develop a way to profile CAZyme content of metagenomic samples without having to do the assembly, without having to do the gene prediction, without having to run them through hidden Markov models. So how we started is we took all of the predicted genes from all of the strains in the strain database that we have. So this is 74-ish million genes and we ran them against the CAZyme HMMs. You see there, a little over 5 million had significant hits against at least one CAZyme, and about half of those had significant hits to multiple CAZymes, and a lot of those were different subfamilies within the same major family. So then using those hits and e-value cutoffs, which is just sort of confidence cutoff for how confident we are a given gene is a CAZyme, we created a bunch of databases with more and more stringent e-value cutoffs, and then we mapped simulated reads from genomes that we knew the CAZyme content of and figured out false positives, false negatives. So on the left column here, you see the false positives. On the right column, you see the false negatives, and then each row is a different CAZyme class. And as we move from left to right within each column, our e-value cutoffs, our confidence cutoffs are getting more and more stringent. And what you see is that false positives tend to decrease as we get more and more stringent, but false negatives also tend to increase as we get more and more stringent. And so what we did is we went through, and for each class, we found sort of the sweet spot e-value where we minimize both false positives and false negatives, and created a new database with these refined e-value cutoffs that's just shy of 1.3 million genes. And then we remapped the simulated reads from the genomes that we had used to decide on those e-value cutoffs, and we also mapped simulated reads from what I'm calling naive genomes that we hadn't mapped previously, and then characterized the CAZyme profile that we predict and compared that to the known distribution. So this is the genomes that I used for what I'm calling training. And the left set of bar plots is the number of unique CAZymes that we're predicting, so sort of a who's there. And the right side of bar plots is the abundance of those CAZymes, so sort of how many of each of them are there, and I've separated them out again by class. And then from left to right within each set of bars, it's what we predict with simulated shallow sequencing, what we predict with simulated deep sequencing, and then what the true distribution is. And what you can see in terms of what we're predicting for who's there, so unique CAZymes, we're very, very close to what the true distribution is from these genomes. On the right, you can see we're off by a little bit where we are over predicting GHs and under predicting GTs a little bit. But importantly, when we map naive genomes to the same database, those biases and sort of abundance are consistent across these different sets of genomes. And again, on the left there, in terms of which CAZymes are there, we are very, very close to what the true distribution is. So it looks like we're doing a very good job of predicting which CAZymes are there, and a pretty good job of predicting the abundances of those CAZymes, regardless of if we had used the genomes to train the database or we're using naive genomes. So this is a different way to look at the same data. So on the left is the naive genomes, on the right is the genomes used for training. And then again, this is the number of unique CAZymes, and you see in almost all instances, we're pretty much spot on with the number of unique CAZymes that we're predicting. And then in terms of abundance, again, you see we are sort of over predicting GHs a little bit, under predicting GTs a little bit. But again, these are very consistent across genome types. So even though there is a little bit of bias here, it's consistent across genome types and is consistent from sample to sample. So comparisons between samples are still valid in this case. And then the last thing I wanted to do was look at the sensitivity of this database. So there's a lot going on here. I'm just going to give a very broad overview. So as we move from left to right in each of these line plots, we're increasing the number of reads simulated from a given genome. And the important thing to pull out of this is that our lines aren't sort of bouncing all over the place. We have a very consistent CAZyme profile that we're predicting, regardless of if we have 10,000 reads from a given genome or 100,000 reads from a given genome, with a few exceptions up there in GH and GT. That's a Pseudomonas strain that we actually get a little bit better at predicting as we increase read depth. So just a brief summary. So CAZymes are carbohydrate-active enzymes. They're involved in all aspects of biochemistry, important in plant pathogenesis, biofuel generation, human health and disease, pretty much anything you can think of where carbohydrates might be important, CAZymes are also going to be important. And then the data I've shown you, I think demonstrates pretty nicely that we've developed a CAZyme database that allows for characterization of CAZyme content from metagenomic shotgun sequencing in a very robust and high throughput way. And with that, I think we have time for questions. If we don't have a chance to get to your questions, feel free to email me. You see my email address up there. And thank you for your time.
Great, thank you so much for that, gentlemen. I do have a couple of questions for you to kick things off with. So to start with, what do you get from the CAZyme analysis that you wouldn't get from standard shotgun sequencing analysis?
Yeah, so I guess at Diversigen, our standard sort of shotgun sequencing pipeline is we give you sort of strain annotations of each of your samples, so how abundant each strain is. And we also do a functional annotation with that. And that functional annotation is based on CAG annotations. And that functional database is sort of restricted to only genes for which we were able to get sort of very confident CAG annotations from. And actually the overlap of those CAG annotations, those CAG genes with the CAZyme database is actually very, very minimal. So it's an entirely different set of functional genes that we're annotating. And in addition to that, with the CAG database, you get like a CAG orthology number, and you get a name like 1,3-diphosphate carboxyl hydrolase or something like that. And you have to like sort of do some digging to figure out, is it a CAZyme? Which CAZyme is it? What's the function of that CAZyme? What other sorts of organisms carry that CAZyme? Whereas with the sort of CAZyme database, you get straight up, this is glycoside hydrolase 2. You can go to the kz.org website. You can look up glycoside hydrolase 2 on the website. It's got all sorts of information about which organisms carry it, what it does, and links to papers about glycoside hydrolase 2. So it's a very different sort of functional annotation than what we normally do.
Great, thank you. And how deep do you need to sequence to do the CAZyme analysis?
Yeah, so I got at that a little bit with that last slide. So for most genomes, if we get sort of about 10,000 reads from that genome, we can pretty accurately profile the CAZyme content of a given genome. And with our sort of standard pipeline, we target about 2 million reads per sample. So 10,000 reads would be about 0.5% abundance in a given sample, and we can fairly accurately profile the CAZyme content.
Brilliant, thank you. Now bear with me for the next one. It's a little bit long, and it's just something that Gerard would like you to comment on. So first of all, he says, congratulations on this innovative and inclusive study design. Then within the same populations, inflammatory diseases, for example, IBD, also rapidly increase over the generations in the new environment. Your work suggests that if we derive patient segments based solely on big microbiome data from homogeneous non-immigrant cohorts, we may end up with models that will perform poorly in the diverse real-world population and potentially exacerbate healthcare disparities. Can you comment on this?
That's a really good point. And, I think one of the major challenges for the entire microbiome field going forward is sampling frame. Who gets in the study and why, and how do you control for potential sources of variation that may be confounders or may just be unrelated to what you're interested in. So yeah, having access to information about someone's immigration status, I think is not something that has been included in hardly any microbiome studies that I'm aware of. Either to perform stratified sampling and using that information, or just to control for it. But it's a really good point that we should be looking at that, especially when we're trying to learn about changes in the microbiome related to chronic diseases that are prevalent in industrialized societies, so that we aren't overlooking unique features that might be present in populations like many immigrant and refugee groups who may be underrepresented.
Great. Thank you so much for commenting on that. Brilliant. Well, that's all the questions we've got. So Dan and Eric, thank you so much for an excellent presentation.
Great. Thanks again.